Right. Good morning, everyone. I just wanted to share this event that's happening tonight, actually. That's kind of a late notice by folks, but they're going to be talking about STEM and at West Valley and also STEM careers. So if you're interested in STEM careers, check that out. I also posted it on Canvas under our events section, which we can go review. We'll go look at that. So welcome. Oh, you can't see me because I have my other screen on. Okay, so I'm going to start by going over the quiz and then we can get started. Today's going to be a lot different than last week, but I'll wait to make announcements until other folks get here. So let's go take a look at the quiz and see how you all did. So here is, did I share this yet? No. So here's today's quiz. Let's go ahead and preview it and we'll take a look and folks can tell me what they think the answers were. So for number one, last week we talked about the metric system. So what are the units for the metric system for length, mass, and volume? What did you all put for this?
C.
It is C, yes. So meters, grams, and liters. For question number two, if we want to convert within the metric system, we would multiply or divide by factors of
10.
Yep, thank you, Maddie. Standard body temperature in degrees Celsius is near. Did anyone get this one?
37.
37, thanks, Anil. The first, so 24, this is near 98 is standard body temperature in degrees Fahrenheit. 24 degrees Celsius is standard room temperature. And then 37 is standard body temperature. The first step of scientific inquiry or the scientific method is to
Make observations.
Yep. So before you go out and start testing random things, one of the best things to do is to make observations. Okay, a hypothesis. Is it a statement of the results? Is it a statement of the predicted outcome? Is it a question you're trying to answer or a statement a scientist is trying to prove true?
B.
So this is B. The first thing is that we don't ever want to make a hypothesis after we've done the experiment. A hypothesis is always a statement. And so that kind of weeds out the other ones. So it's your prediction of what you think is going to happen. It's your guess.
Can you explain why it's not A?
Sure. So a hypothesis, a statement of the results of an experiment. So that's telling what happened. So we don't know what's going to happen yet. So a statement of
D.
Pardon me?
I actually meant D, sorry.
Oh, D. So we never want to be trying to, quote, prove it true. It's true that scientists may want their hypothesis to be correct. If you go back and look at the 5 guidelines for a hypothesis in the lab from last week, one of the dangers of trying to prove a statement as true is that it can lead to bias in science. So it's your prediction of what is going to happen, but if you're trying to prove it true, that can lead to some really shady science projects, science experiments. Does that answer your question?
Yes, thank you.
So when we add a control and when we add replication and all these things, this is trying to make sure that we're not putting that bias in and trying to prove it true. And when we finish with a hypothesis, if our results support our evidence, if the results of our experiment support our hypothesis, we still don't say that our hypothesis is true. we would say that the evidence supports that hypothesis. We would accept that hypothesis. So science is a process. And so you'll rarely hear scientists say things really definitively, because they're always saying like the evidence shows that this is this way, because we're still learning. Okay, in our experiment with yeast and different drink sweeteners, which liquid represented the control setup?
Water.
It was water, correct. The other ones would be the experimental setup. In our experiment with yeast and drink sweeteners, the amount of gas produced by the yeast is the control, the independent variable, the dependent variable, or the replicate?
Dependent.
Dependent variable. And if anyone had problems with that, remember, the independent variable is the thing that you change. The dependent variable is the thing that you measure, or the result of that change, okay? The factors that you want to keep constant during an experiment are dependent, standardized, independent, or experimental variables.
B.
Pardon me?
B.
Yeah, B. So standardized variables are those ones that you want to keep constant so that you're not changing things between different setups. So last week, we had the control, and then we had the different drink sweeteners. And what are some of the things that we kept constant between the different bags?
The measurements, like the
Yes.
amount of yeast that was in each bag.
Yes, the amount of yeast, the amount of liquid, how we rolled the bags. If you did them all different, that could lead to different results. You put them all in the same temperature of water. So that would be another thing that we kept standard. So you'll see this as we go along, that as we design different experiments together, next week we're doing an experiment together, you want to keep all variables in the background the same if you're going to be changing another thing, so that you can get a true snapshot of what's really going on. So here's an application. These next questions are applying the independent dependent variable. So you've seen more hummingbirds coming to your yard. You decide you want to put food out for them, but you don't know what they like. So you put out water. You put out that red nectar water that you can buy at a bunch of different hardware stores and online. And you put out your own, sorry, big truck going by, your own homemade sugar water mix. And you want to determine which one they like the most. And so you're going to look at how much liquid is disappearing, and also look at which one birds are visiting the most often. So in this experiment, the type of liquid you're putting out is which type of variable: the dependent, the independent, the control, or the replicate?
Independent.
Thank you, Neil. Yes, it's the independent variable. This is the thing that you're testing. It's the thing that you're changing. So the type of liquid food you're putting out is your independent variable. Does anyone have any questions on that now that we're starting to apply these to new concepts? Okay, when you measure how much liquid is used by the hummingbird, this is the dependent, independent variable, or the replicate, the control, or the replicate?
Dependent variable?
Yes, thanks, Paul. The dependent variable, this is the thing that you measure, okay? So we're measuring how much liquid is disappearing, so that not disappearing, being used by the hummingbirds. And so that's how we're measuring which food they might like the best, okay? Now, if we wanted to improve the results of this experiment or any others, which one of these could we do to improve our experiment? Would we increase how many times we repeat the experiment? Would we decrease the variables that we want to standardize? Would we increase the number of independent variables, the thing that you change? Or would we decrease the number of samples for each test?
Increase the number of times you repeat the experiment?
Yes. So if anyone puts C, The independent variable is the thing that you change. So for instance, for last week, if we were to put different drink sweeteners and then put those bags in different temperatures of water, that's two different independent variables, and it doesn't, that's just giving you completely random results. You don't have a good test because you have multiple independent variables. When you're doing an experiment, you want to have change one thing at a time. So if we wanted to know why wildfires in California are increasing in frequency and becoming hotter, we wouldn't want to measure, we would want to measure one thing at a time. Is it, so scientists are looking at climate change. They're looking at the buildup of dead materials that haven't been burned over time. They're looking at the decrease in water. So we look at one thing at a time, and then we can put all those results together to find something that resembles the truth. Here's this one I'm going to grade independently. So this is that hummingbird question again, and you were just asked to write a possible hypothesis. So all I'm looking for here is that you had a statement, not a question, and that you based it on the set of parameters you were given, okay? So I'll grade that one separately. And two ways to display and analyze the results of an experiment are
A graph and a table.
a graph and a table, okay? If you put something like that and you got it wrong, it's probably just a misspelling, and I'll be looking at that as well, so don't worry about that. So any questions about this? Okay, so we're going to finish early today because you're going to be able to do a lot of this experiment on your own. And so if you have other questions, you can stick around and we'll go over them together. So today is kind of special because obviously we're not in a lab working with microscopes. So what I've done is I've put together a bunch of images of my microscope images and also videos for you to look at, to learn about microscopes and their application. Some of you are interested in going into the fields of science, so this will give you a background in how microscopes are used, which type of microscope you might want to use for different applications, and then also what you might see under a microscope. So for all of you, getting to see what the images that a microscope kind of unlocks for us is really special, what we've learned from microscopes. So what I'm going to do today is introduce the lab and kind of walk you through what your part is. And then you can do the lab on your own. Now you're welcome to work with someone else, but it's a lot of like watching videos and looking at photos and drawing. So it's not really necessary for you to work with other folks. And the other thing is, the sky is blue here today. And I talked to my friend in Palo Alto, and the sky is blue there today. And I just want to encourage you all to enjoy finally a day where it's not brown or gray or orange or white outside. So what color is the sky where you all are in the chat?
Low key blue.
Gray again.
Oh, you have gray?
It's still gray.
Blue, mainly blue.
Okay, good, good. We've got lots of blues and one gray. So I just want to encourage those of you that have good air quality to go enjoy it. I think it's really hard on mental health when we already have to be on the computer and we can't go to school, and also we can't go outside. So I just want to, if you want to do this lab later, you can stay for the introduction and the announcements, and then you can do this whenever you want. It's just due next Tuesday. You will need your lab 3 that's in your lab manual, and if you don't have that, I will be posting a Word and PDF version. This will be the last week that I do that because you should have ordered your lab manual by now. So let's go ahead and jump into today. I just want to see, a couple announcements. We still need a note taker for this class. For those of you that need a note taker, I am posting the PowerPoints and I am posting study guides. So that's sort of trying to help meet that need. But we don't have a note taker for this class, and we have a couple folks that need one. So if anyone's willing to do that, a reminder that you get priority registration. You get a letter that you can submit when you apply to a college, and you get a $35 gift certificate to Barnes and Noble. So if you're interested in helping out with that, you could just take notes by hand and then type them, or you could type them as we go, or you could write them on your lab manual and take photos of that. There's a lot of different ways to do that. So whatever works, you'd be helping out a couple folks in this class. So let me know if you're interested in that. Okay, so so next week, another announcement. Next week, just a reminder, in your lab kit, there's this piece of paper. Look at this ahead of time because it tells you which materials. This came with your lab kit. It just has a list of materials. And check this out because it's going to give you the things that you need to pull out of your lab kit. And there's also a list of things that you need to gather from your house, like tape, scissors, a permanent marker, 3 containers for different temperatures of water. So go ahead and get this ready ahead of time. Next week, we are doing a hands-on lab again, like we did last week. It shouldn't be as long as a lab as last week. We kind of went right up to the end, and my goal is that that doesn't happen as a norm, but just heads up on this, okay? Other announcements? Did that? Okay. Oh, grading. I'm a little behind, but I'm going to finish that up today, so all of your grades will be up to date later today or tomorrow, okay? So let me go ahead and share today's PowerPoint. And actually, let's go to Chrome first. So I want to start with a video. This video is kind of brings together last week and this week. So last week we learned about scale, and microscopes have really helped us learn about scale. So imagine a time when we didn't know what a cell looked like, when people did surgery without washing their hands because we didn't know about bacteria. You know, microscopes have completely revolutionized what we can see. And so I want to show this video because it shows it's going to zoom way out, because telescopes have taught us about the scale of the universe, and then microscopes have taught us about the scale of the inner verse, okay, the inside of ourselves. And telescopes and microscopes are really just about lenses and mirrors. So lenses and mirrors, how they're formed, how they're shaped, has everything to do with how this works. So this video is just to give us an idea of scale. Sorry, just one thing. If you look at the bottom here, you can see what scale we're at. So right now we're 10 kilometers away from the beginning of this video. So you'll see the scale here change. So obviously not all of those things can be seen from, stop, can be seen just through microscopes and telescopes at the far limits of each end. We have to use different kinds of imagery and use different rovers and different things we send into space. And then with physics, it's a whole different thing with how we know what the nucleus looks like and what the different atoms of the nucleus look like. But as far as microscopes go, we've learned a lot just from the use of these lenses and the mirrors. So I have some photos to start us out with just to kind of get excited about different things that we can see. This is the foot in parentheses of a barnacle. So if you all have seen barnacles, they actually stick out this bristle-like structure to feed plankton out of the water column. And I talked a little bit about what was life like before microscopes when I was talking about things like bacteria, and we didn't know about what a cell was, so there was no such thing as cell biology. So really, microscopes revolutionized the field of medicine, and they also revolutionized the field of discovery. We didn't know that there were little plankton and algae in water, so waterborne disease and things like that. We didn't know about any of that. So microscopes really opened up a whole new world. And microscopes actually started out as a toy and were kind of used not very much by scientists. It wasn't really a great tool for a while until they became until people started kind of honing in on making them have higher powers of magnification. So here's some more photos. This is a photo of a plant cell. There's this whole new world where people are doing art, microscope art. And so these are actually artists who have taken these photos of these different pictures and released them. This is, any guesses what we're looking at here? If we're zoomed way in, what might this be that we're looking at? Sorry, go ahead.
Fish scales.
It really looks like fish scales. It's very similar. It's a butterfly wing, which has a very similar look to it as fish scales. Awesome. And you all can also put guesses in the chat because I have a few more guesses that we're going to do here. Pull up the chat so I can see it. A second. Okay, so the next one. These are some, in plants, you can actually add different dyes or use different types of light that make different proteins glow fluorescent. So you can see those here. These are also those same similar properties where it's a protein that under different types of light, they glow different colors. So scientists will use this in research to learn about different types of proteins and how they're arranged. These are urea crystals. Urea is what is in urine. And so this would be an animal's urine crystals. So even pee can be beautiful. This is an old plant. So we're actually going to look at living plant tissue this week. Have you all started talking about plant versus animal cells in lecture yet?
Like prokaryotic cells versus eukaryotic cells.
Okay, so you haven't done plant and animal yet. One of the big differences between plant and animal cells are these, and some of you might remember this from bio in your middle school. What do plants have around them that doesn't? So this is a dead plant, and this gets left behind. It takes a really long time to break down. Any guesses what this is?
A cell wall.
It's a cell wall, okay? So even old plant tissue sticks around for a long time. This is why logs and leaves take a long time to break down. This is the cross-section of a rat brain, just to show you another image that has, a lot of, for better or worse, a lot of research happens with rat brains to learn about different human conditions. And this is a visualization of that, looking at those fluorescent proteins again. This is the mouth of a moth. So this is called the vampire moth here. And they have these scraping bristle-like structures for feeding. So this is a zoom in on the mouth of a moth just to see, so you can see how crazy and complex that is. All right, any guesses what this is? This is a real thing. I'll give you a hint, it's part of an animal. And you can put guesses in the chat. This is a low stakes question if you have a hypothesis of about what this is. No guesses. So this is actually the foot of a beetle, okay? A foot of a bug. So you can see the leg of the beetle here, and they have all these structures here. So you all have seen beetles can climb up the side of a wall or the side of a tree or the side of a slick surface, and it's because they have these crazy feet. And yeah, they're pretty too, Alexis. So really remarkable to be able to see the bottom of a beetle foot.
Are those like suction?
Yeah, totally. Yeah, these right here are little suction pieces, and here as well. And then it has the bristles also. So it's got multiple ways of kind of holding on to a surface.
Does that like count towards other insects that can be, how you call it, be firm on a surface that's like vertical?
Yeah, they'll either have, exactly, yes. So it helps them hold on to those surfaces. So, you know, you may have wondered how they could just climb straight up a stem or straight up a leaf and not fall off. And a lot of them have these different types of structures on the bottom to grip. And this one's a lot larger because it is a beetle. So they're supporting that big weight. So they have a lot of different suction surfaces. This is a fun one. Any guesses what we're looking at here? It is the tongue, Elizabeth. Yeah, this is what your tongue looks like up close. So those little bristly things that you can feel on your tongue, that's what you're looking at here. So you can see also these light white pieces here. These are dead skin cells. So our skin is constantly sloughing off and renewing itself. And so you can actually see that happening here. You can't see any taste buds here. Taste buds are round and more towards the back of the mouth, but this is what your tongue looks like under really high magnification. This one is kind of gross. I'll just tell you what it is. This is what eyelashes look like. So again, this is super zoomed in, so it may gross you out to see what this looks like. And I know that the colors are green, but that's just because of a stain that's used. But again, you can see the skin is kind of sloughing off. You can see the hair follicles coming out. So I kind of like the gross stuff. So here's one for those of you that may like that too. Okay, so there is one cell in the human body that is visible with the naked eye. It's the only cell that's big enough. It's larger than 100 micrometers, which is pretty small. But we can only see things that are larger than 100 micrometers. Micrometers is another way to say that. So any guesses what cell in some humans we can see with the naked eye?
Is that like the human egg cell?
It is. It is. It is. So that's why I said some humans. So this is an egg cell. So that's what that looks like. So those are just some pictures to kind of get you interested in some of the awesome things that we can see and what we're learning about through microscopy. So as I said, the microscopes were started in the late 1500s as a toy, and they told us a little bit about blood circulation, but eventually, as you'll learn in the module today, you'll learn about the person who invented a microscope that had a higher magnification and allowed us to see cells for the first time. And the cool thing is he wasn't a scientist. He was just a person that was curious. So I always like to remind people that you don't have to be a scientist. You don't have to go and get a PhD to have questions and discover the answers. And in fact, a lot of things have been discovered by people that weren't trained scientists. So This discovery gave us a lot of understanding about things like tuberculosis and cholera, which were the two big causes of death at that time. So again, microscopes changed medicine and health. So here's some pictures of bacteria up close. These little swimmy things are called flagella, if you all don't remember that. So this is E. coli, which is one of the most, E. coli and Salmonella are the two that are the most, oh, I'm sorry, this is Salmonella, E. coli are the tiny dots. These are the two that are the most common that make people sick. Salmonella with things like chicken, and E. coli comes from the waste of any animal that gets into a water or food source. This is what staph looks like. Staphylococcus, it's an infection. If any of you have ever had staph infection, it's pretty nasty. And staph can invade your skin and it can invade your body. And this is one of those that you've probably heard about, how a lot of antibiotics are becoming not strong enough for certain types of bacteria. And this is one of those. This is the staph that has become resistant to a lot of antibiotics, which is really pretty dangerous. Here's some viruses. This is what HIV looks like under what's called a transmission electron microscope. Scanning and transmission electron microscopes use electrons and light to be able to get images of very tiny things. And there's a video in today's module that talks more about electron microscopes. But I've only seen one electron microscope in my years as a scientist, and they're extremely expensive, but the things that they can show us and tell us are really rewarding and interesting. So today, let me go ahead and get out of here because we are not doing all of those things today. What we are going to do today is different because obviously we don't have access to microscopes. I do want to talk really quickly about this top module here, just as a reminder. I have some, I need to get rid of this pre-labs because you don't need to do pre-labs, but this is our fall schedule. These are the assignment and quiz dates. Here's student resources if you end up having any needs, like for food, financial aid, mental health, or if you need support with test accommodations or note-taking accommodations, that's the Disability and Educational Support Program. But I'm updating this section regularly. This is all things related to science. And so if you are interested in pursuing a field in science, we have a Women in STEM Club for the first time this year. So anyone that identifies as female can join that. This is a Friday. We're having a regular speaker series. And I'm going to go ahead and open this one because we have a really awesome speaker this week. So Reagan Beers is coming to talk with us. This is Reagan. And she'll be sharing her experience. She works for the National Renewable Energy Lab. And so each week we're going to have a person who has either graduated from West Valley or has some connection to West Valley to come and share their experience, their career experience. And so this is this Friday at 1 o'clock if you're interested in a STEM career. And then I just posted this as well. Tonight is a back to school night for folks interested in STEM. And to remind you what STEM is, STEM means science, technology, engineering, and math. So if you fall into any of those categories, or if you're just curious, STEM curious, then you can go check that out. Okay, so this is this week's module. I put the presentation I just did here there. So there will not be a quiz next week. Dun, dun, dun. Because there's really nothing for me to test you on. Today you're doing a lab, or whenever you decide to do it, you're doing a lab where you're going to be looking at videos and looking at images and drawing them and making observations, okay? So I'm gonna walk us through this. If you want to get the points so there's 5 points here embedded within this lab, and that will go towards the entire lab. I'm going to make the lab worth 15 points. And then if you want the remaining 10 points, I'm going to post an optional activity for you to do that's related to looking at scale and making observations. So you're going to make observations at 3 different scales. So it's optional. And I'll put that up today right after I finish going through this. So no quiz next week, optional activity for you to get those 10 points if you want them. You will not be penalized if you don't do it. Any questions about that before I go through the module or any of the announcements? Okay. So the first portion of this is, it says quiz, but it's really just, the reason for this is so that you can go through and answer questions. So you're going to be watching this video, which is an introduction to the history of microscopes. And then you're going to answer these 3 questions. And you have 3 attempts. So if you don't get all 3 right on the first time, you can do it again, and you can do it again, and it will save your highest quiz. Okay? So that's the first thing. The second thing is learning about microscopes in general. So there's 2 different videos here to watch. One is comparing, in the lab where we're going to be looking at images, and we would have been using 2 different types of microscopes. So we have dissecting microscopes and we have compound microscopes. The dissecting scopes allow us to look at things 3D. They allow us to look at larger things like a leaf, or like a bit. You could look at the entire leaf. You could look at your hand. You could look at a bug, like you could put the entire bug or the entire flower on the stage, and it allows you to look at it because it doesn't zoom in as much and there's a lot more space. If you were going to dissect an animal or a plant, you would use this dissecting scope because you can do it and have a really clear image. The other type of microscope is called a light microscope, and this is for things that we want to look at that are very, very small, like blood cells, or you know, when people do counterfeiting of bills or something like that, you might use a compound light microscope. A lot of the forensics uses compound light microscopes. So for those of you that might be interested in forensic science, then that's what
Hello, I'm scared for a second.
Anil?
Yes, ma'am.
Anil, you're not muted. I don't know if you were saying something, but so you're going to watch these 2 videos and as an introduction. Then the rest of the lab is looking at images and drawing those images in your, what is my cat? Oh God. My cat brought a gopher into my house and it's still alive. Okay.
He just wanted to bring you a friend.
Yeah, he's
New
my
friend.
cat. So my cat has brought into my house and eaten 14 gophers this year.
Oh no.
That's good, actually.
Yeah, no, it's awesome. I have a date. I'll show you. I have the data for it. And maybe we can go look at my cat really fast. This is biology in action. Let's see. Here's the cat with the gopher. Can you see it? Can you all see that?
That's a nice tile floor.
Oh, you can't see him yet?
There we go. Now I can. Oh, yeah.
And then this is my data point here. Can you
The
all
kill
see that?
tally.
Yep. So we're at 14, and then there's other, just to show you that I'm still doing data collection, even in my own home, in my biologist boredom. Okay, well, that was exciting. Okay. So, woo, okay. He's gonna eat the whole thing, you all, from head to tail. I've watched him do it. It's disgusting. And you can hear it too. So anyway, getting back to our lab manual. So in your lab manual, there's a bunch of different places to draw things. And so you're basically going to go through, and I've told you on which page you're going to draw each portion. So I have pictures of the different organisms that you will need to be able to draw this. There's a lot of sounds coming from the other room. Okay, so that's what you're going to do to complete page 3-6. And I'm going to list, you're only going to be submitting 5 pages this week. And so I'll list that on our Canvas page. The next page, you will be looking at the compound light microscope. So this is the one where you're looking at things up closer. And I discussed the concept of what's called depth of field. So if I were to put a leaf on a compound light microscope, which you'll know more about the different types of microscopes after you watch the videos, you can focus up and down through the leaf, and that's called depth of field. And I have this photo here as an example because for those of you that like to take pictures, sometimes it's impossible to get the entire thing in focus, and it's the same with a microscope, and that's depth of field. So if I want to get these in focus, I have to change where I'm focusing in the depth of the image, of the thing that I'm looking at, okay? So you're going, here's some examples for you, and then you're going to be using these two images to answer the questions on page 3-9 and 3-10. So basically for most of the rest of the lab, you are going to be answering questions based on images. So that is for page 3-9 and 3-10. There's lots of sounds coming from the other room. Okay, he's chasing it and throwing it around. I might show you all again. This is part 5 where you're going to be looking at what happens to the letter E. With the dissecting scope, which is the one that you're using for 3D images, when you look at an image, it's a true image. But with a compound light microscope, when you look at an image, it's actually a reverse of the image. The mirrors and lenses end up flipping the image so that you're always looking at a reverse of what is truly on your slide. So you're going to learn how to prepare a wet mount, which is how we make a slide. Sometimes when you're in a lab, you use slides that are prepared ahead of time, and what they do is they use chemicals to fix it, and then they seal it off, and then those are good for years, sometimes 10 years. 10 years at a time. He just knocked over a chair. And then here, you're going to use these letter E's to do some drawings within your lab manual. And so all of this is pretty self-explanatory. The next thing you're doing is looking at a leaf, and I introduced talking about leaves and the different organelles of leaves. These are 3 different magnifications of what this Alodia is. Alodia is an aquarium plant, if any of you have an aquarium at home. And this is what the Alodia looks like on a slide. And these are 3 different magnifications. So we're at 40 times. So we're looking at 40 times large, more magnified than the naked eye can see. This is 100 times. This is 400 times. And again, you can see the cell wall. What are these little green things floating around in here? In a plant cell. Oh God, he brought it in here. Okay, so maybe you can see him. Snuffy, can you all see that?
Oh yeah.
Again, he
Oh,
just
that's
wanted to
good.
bring you a friend.
Yeah, he's brought me a lot of friends. Get it? No, don't do that here. Oh gosh, okay. This is Zoom. This is the Zoom live. Okay. So
I just changed my mind to get a cat.
it's kind of awesome because I have a big yard and the gophers eat my plants, and
Pest
he
control.
eats the gophers. So
control.
I have a whole cycle here going on, and I don't like the gophers because they eat my plants. So he can eat the gophers, but I don't really want to be a part of it. I don't want to watch it. I don't want to hear it. Ugh. Okay. This is the leaf zoomed in at 1,000 times. And this is the video, so you can see what happens in the leaf, what you would have seen under the microscope. And then the last part is looking at other specimens. So you're going to pick 2 of these to draw. This is a really cool one. If any of you have tattoos, when you get a tattoo, they inject the ink below. This is the skin surface right here. And they inject the ink below, and this red part here is the ink of the tattoo. So you can pick any 2 of these images. This is a dollar bill. Dollar bills are made out of cotton fibers, so you can actually see the fibers of cotton. And here's some other, this class really loved playing SpongeBob SquarePants. So this is the actual character that plankton is based off. This is called a copepod, and you can see the antenna and the eye. So that's a real thing. So that's what you're doing this week. You're only going to submitting, I was wrong, you're submitting a total of 6 pages. And so I will add that part to the module for the submission portion here.
Do
And
we
I'll-
draw the images on a separate piece of paper or do we have to?
You can. That's a good question. So if you would prefer to, you all could actually draw all of the images, because most of what you're doing today is drawing. You could put all of the images on a separate sheet of paper and then just take a picture of that and submit that. Or you can draw them within your lab manual, and then you take a picture of the 6 different pages that have the drawings on them, and you can submit that. So it's up to you. That's a good question.
And then do we have to take notes on the videos or could we just watch?
You can just watch the videos, and the videos you will need to watch them so that you can really understand the difference between the dissecting and the compound light microscope, and to tell you a little bit about how they're used, but you don't need to take notes. It's to help introduce you to what you're going to be seeing. So it's going to help you understand how the microscopes are different.
For the drawing part, can we do them on the, how you call it, the paper, the worksheet?
Yes. Yes, within the module, I tell you what page to do the drawings. So you can either do it on the lab manual, the worksheet that you have, or as Paul asked, you could do it on a separate sheet of paper. It's up to you. And I want to talk a little bit about drawing, because I want those of you that, a lot of people have an anxiety about drawing, and I just want to talk a little bit about that, because when we're drawing in this class, it's not about whether you're a good or a bad drawer, person that can draw. It is about whether or not, I have to leave because I can hear the crunching. Yuck. The cat eating. Let me go ahead and stop this so I can see you all. It's not about whether or not you're good at drawing. It's about being able to record information. Okay, so think of drawing instead of as a beautiful piece of art. In this case, think of it more as scientific illustration. It's really all about recording information. Okay, and using color is actually really helpful. So if you want to capture the details, you might consider using color pencils or markers. Color pencils tend to work better. It's not required, but it will allow you to have a little bit more detail. So just think about this as practice, because we will be drawing other things in the future. And I'm really just looking for details on the drawings, okay? So, and the other thing about drawing is, you know, if you're drawing a picture of, here's a picture of some sea urchins. And if I want to zoom in on a sea urchin, I don't want to draw it this big. I'd like you all to try, there's circles in your lab manual that are about this big. When you draw, try to fill the circle rather than make these little tiny little drawings, so that you can actually record the detail, okay? So that's what we're looking at for drawings, okay? Questions? So really, this lab is about looking at images, looking at how microscopes are used, learning the history of microscopes, and then using that information to record details, okay? So I'm gonna post 2 things. I'm gonna post the optional activity for 10 points. That's 3, it's gonna be an additional 3 drawings, but of something around your house or something outside. And then I'm going to post the submission where you upload your drawings, okay? And again, there's no quiz next week. And you can assemble your materials ahead of time using the list in your lab kit. Okay, Alexis asked about the 6 pages for an assignment. The 6 pages are the 6 pages of the lab manual where the drawings are. And that's all detailed in today's lab. As you go through, it'll say, look at these images, and on page 3-6, draw what you see in the two bubbles, okay? Or you can do what Paul said, and you could put them all on one sheet of paper, on 2 sheets of paper. So there's 6 pages from your lab manual that you can do your drawings in, and then take pictures of that and upload that. Or you can do them all on separate sheets of paper. Okay, just make sure that you label everything.
But then on 3-6, it says like about obtaining a patch of Velcro and then rough side dissecting microscope.
You don't have to, there's a picture of Velcro in the module. So you're
Okay.
not gonna be, don't use your module, your lab manual as a guiding force. Use the module as your guide, okay? Because there's so many things in today's lab that we can't do. And that I'm not gonna have you memorize the parts of the microscope because I think that's ridiculous if you can't see it and you can't touch it. If you stay in the field of science, you're gonna get that opportunity again. But I don't think it's relevant for you all to learn all the different parts when you can't even see the microscope. So I'm not requiring that. So
And
there's
then
a lot of parts in today's lab that you're not going to do. So follow the module step by step, and it will tell you what parts of your lab manual to fill out, or you can submit everything separately.
And how are the labs graded? Is it graded by like completion, or is it graded on like you doing like in the sense like if all the answers are correct?
Both.
Both? Okay.
So first step is completion, and then I do grade for correctness, and I know that
Quality.
in a lot of cases there aren't, this is science, so there's lots of different possible answers. So sometimes I won't even take off points. I'll just say, oh, actually, I'll just leave comments for things that might be misunderstandings. So I'm looking for effort, and that you've completed everything, and that you've, a lot of things we go over. So if we've gone over them at the beginning and at the end, this doesn't apply to today, and you still aren't getting it, it means that maybe you weren't here, or maybe you weren't really here. I see black boxes, and so I'm looking for that you all have put in the effort and are trying your hardest. So.
No problem.
Other questions? Okay, so I'm going to stick around for folks that have other questions, but I'll go ahead and right now I'll put up the lab where to submit your lab, and more detailed instructions just about what to submit. But it sounds like we feel like we get it, and I'll be here again. No quiz next week, and you can do the supplemental activity if you'd like. And if you have any questions about the STEM events or anything like that, I'll stick around too. So otherwise, you all go get some blue sky today and get outside a little bit.